Okay, can somebody please confirm you can hear me?
I can hear you.
Thank you. Before I share my screen, let me talk a little bit about the grading. Let me go to the grade book. I got lab 00 and lab 01, meaning the first two labs of week one, graded. I also got worksheet for lecture one or chapter one graded as well. I normally talk about the grading in the lab, but today I'm going to do it in the lecture. And I have started grading the extra credit study sheet, but I've only gotten a few students done. And that's about it. I do have the extra credit study sheet to finish, and then the extra credit picture. Let me see if the extra credit picture is due. I think it's due, yeah, next Saturday. So I'm not going to get that one graded. And then, of course, I've graded the environmental quiz and then the plagiarism quiz one and the plagiarism quiz two. I did state that students can resubmit those quizzes for full credit if you get it done by the due date. I think the environmental quiz is either due tonight or tomorrow night. Let me look that up. And then the plagiarism quiz, I think is due this Saturday. You must get 100% on both the plagiarism quizzes outside of the first question. If you miss the first question, I won't count it against the 100%. But if you miss any other questions, then you will not get any points on the 100-point infectious disease project. So make sure you get 100% on the plagiarism quiz questions, at least questions two through the end of the quiz. And that should be pretty easy because I'm giving you the answers. I'm looking to see when the quizzes are due. Just a minute here. Oh, come on. Yeah, plagiarism quiz two, and I assume one is due until January 17th, the until date. And then the environmental quiz, the until date is January 14th. So tomorrow at 11:59 p.m. You have time to get that. Let me state on the environmental quiz, only about 25% of the students showed me their complete environment. And I was a little surprised because this time I specifically stated what I wanted you guys to see and then suggested if you don't know what you're showing me, then show me more than you need to. And uh, the uh, the success rate dropped from about 50% to 25%. So I was wondering if anyone could mention something about why they did not succeed in showing me their environment around their computer when
My difficulty is just not being able to see what I'm showing you, which I know you just said show more than what you thought you would need. And I thought that I did show more. I even showed upwards too. So I'm wondering if there's a way that like there's a mirror option to be able to show like what we're showing you.
yeah, no, there isn't a mirror option in Lockdown browser, so sorry, I
Darn.
can't make it available to you. And I assume the reason is that they don't want anyone being able to see the recording other than me, but I don't know. Anyways, there is no mirror option, and all I can suggest is you have the camera or you think you know where the camera is, just show me more than what you think is necessary. Like I stated, show me before where the table is or your desk or where the computer is sitting. Show me where the computer is sitting, and then show me behind where the computer is sitting. And then when you come to the end of the table or desk, go further. Okay, and then you'll hopefully get everything. Last term, I had a student who every time she showed it to me, she said, "Well, I can't see what I'm showing you, so I don't know if I showed it to you." And every time I said, "Well, show me more." And she never did. So I don't know what to do. Like I said, this time I've written out exactly what I want. And if someone can suggest wording that will help me. The other option I have is in future terms, I can just make it so that students come in and then they take a written exam at Clark College. I can make that happen. And I
I
have to
have
say that if the percentage doesn't go up from 25%, I will probably in the future, not this term, but in a future term, just make it that students have to come in to take the exam on paper and pencil.
my only suggestion would like request would be if possibly, do you think you could like record yourself doing what you would want the students to do so that we have like a visual with your own computer? Because I know you tried to do it on the first lecture, but your background was on, and so it was we couldn't see what you were doing specifically. So I wonder if there's a chance you could just record yourself doing what you would like us to do.
I can't use Lockdown Browser to do that, but I suppose I could do it with, um, what the heck is this program called? The Zoom. And then I can just make it a separate meeting and record it and then make that available to you. The thing is that to show it, I have to remove the background, and I can do that. It isn't easy to do. And then I have to reinstall it after I do that. But like I said, I could do that and try and have it recorded. But like I said, what you want to do is you record before where you think the computer is sitting, record where the computer is sitting, and then record after where the computer is sitting. I need to see the entire desk or table where your computer is sitting or where the keyboard is sitting if your computer is up in a on the wall, for example. But um, anyways, just show me the complete table or desk. Oh, let me state that Lockdown Browser recommends putting the computer on a hard surface. Occasionally, I get students putting it on their bed or putting it on their lap. And I don't know why, but Lockdown Browser suggests not doing that, that it fouls up Respondus Monitor recording the session. Okay. So, um, anyways, they want it on a hard surface. Any question about any of that? All right, let me get out of the grade book. Okay, go on.
The only thing I was gonna add in is so when I retook the plagiarism quiz, it took a while of me not being like in the monitor, but after a while, when I would peek at my laptop screen from behind the computer, it actually popped up a box and was showing what I was showing you. So I was like able to see if I was getting the whole table in. But it took like at least a minute or two for that to come up.
Can you write down exactly what you did so you can see that? And then I'll make that available to students because
I
I,
I
I
think
don't know
it's
how you did it.
I think it's just a feature of the lockdown browser response monitoring. I don't know. It's like as if you're not in the view of the camera for like a specific amount of time, it will like start like screen sharing what it's looking at on your screen. It That's what it did for me.
All right. Thank you. Like I said, I didn't know that, but um, all right. Um, all right. Any questions about any other topic before we start the lesson?
Are the videos usually available to us on YouTube? Because I was not able to attend the meeting on Thursday, and I was like traveling, so I couldn't access it even like on my phone. So I wasn't
Let
able to
me
complete the lab
see.
after that
I
because
know Tuesday got up. Let me see to make sure I actually got
Thursday did
um,
eventually get up, but
Thursday up. Let me check and see
I
if I did.
the lab assignment was due before I could access the video. So yeah, I just kind of had to figure things out, and then I watched the video after it got uploaded, but it was after the assignment was due.
Uh-huh. I have the lab from 1/8. Does anyone know, is that Tuesday or Thursday? Let me look at the calendar here. Yeah, that's Thursday, so everything's up. Like I said, if a day later it isn't up, send me an email as a reminder and I will get it up. But this time it looks like I got it up uh, shortly
When
afterwards.
I
Sometime on the 9th, I got it up.
Okay, yeah. So was the assignment due on the 9th? Like the lab that we you went over in the um Zoom on
Oh, you're talking
Thursday?
about the lab one? It was due Friday, yes.
Yeah, so it
Because
was
that's the
due
first
Friday
week.
at 8 a.m. and I did not have access to the Zoom recording at that
Uh-huh.
point, so it was harder for me to answer the questions. So I was just curious in the future if it'll be possible to upload the recording sooner, if it's just like the process of uploading them that is not
Yeah.
possible to upload them
The
quicker.
problem is that once I upload it, it has to put in the subtext, and that takes time. And this became available January 9th. I'm not sure what time it became available, but let me see if I can find that out. But I don't remember when I got it loaded up. But usually I get it loaded up, about 10 to midnight. And then it becomes available like an hour or two afterwards. But my apologies. Like I said, that should only be a problem on the first, actually for only lab one, because it's due Friday at 8 a.m. And
Will most
hopefully,
assignments in the future be due like Friday
yeah,
at
the
midnight
assignments in the
then?
future will always be due at 11:59 p.m.
Okay, it's
Saturday
just that
night.
first week for attendance, correct?
Yeah, that first week I had to send in who was attending my class by 5 p.m.
So
Understandable. Thank you.
so I
Okay.
will
But in
have
the future, if you have a problem like that, just email me and I can postpone the date that your lab is due.
Okay, perfect. So I will be able to access the recordings before the due date
You should
next
be
week
able
or
to.
yeah.
Like I said,
Okay,
if
perfect.
if there it isn't up 24 hours later, just send me an email as a reminder to get it posted. Because occasionally I forget, but most of the time I send it up um like between 10 and midnight
Okay,
of the day
perfect.
I make
Thank
the
you.
recording. It does take time for the recording to finalized too, but it's usually recorded in, I don't know, 15 or 20 minutes. So I can put it up 15 or 20 minutes later, but I don't usually get around to doing it until 10 or midnight. Okay, sorry about that. Like I said, if there's a problem on the lab, um, just let me know and I will allow you to make a correction on that. All right, let's go on and talk about today's lesson. Let me share my screen here. Optimize for video sharing. Should be on my screen now. All right, today is January 13th. Boy, that took 15 minutes of time. We're supposed to be talking about chapter 1 and ending chapter 1. We're a little behind schedule and beginning chapter 2. Let me warn you, we are having our first real quiz. It's a practice quiz, so I guess it's not a real quiz. Practice quiz, it's either 00 or 01. Let me look on that. It's actually practice quiz 1B, requiring respondents to lock down browser and webcam. You will get extra credit points for doing this quiz, but you should take this quiz because it will give you some idea how hard my questions are and what I expect from my students. With this quiz, if you don't get the questions correct, you can take the quiz a second time, and this will be the last time you'll be able to take a quiz 2 times. I don't tell you what the answers are, but I tell you which questions are wrong. So if you got a wrong answer, you can make another guess. And each question will be worth, I think it's 0.1 point. So this will be giving you extra credit point, and we'll give you another chance to practice sharing your environment around your computer. And let me state again, please show the complete table or desk. You won't get the credit unless you show me the complete table or desk that your computer or keyboard is sitting on. Any questions about the extra credit quiz? It's due 11:59 p.m. January 14th. That's Wednesday, same day as a normal quiz. Oh, I don't. Oh, yeah, that's right. Never mind. All right. Anything else to talk about that? Oh, excuse me here. Ah, don't know where that came from. The practice quiz is going to be over chapter 1, lab 0, lab 1, and the material I covered in the syllabus. There will be a lab today. Usually I do sneeze twice, by the way. All right. Scroll that down. So I have that we were discussing the 3-domain system last time, and that we said that each domain has a cell type or a superkingdom. So domains, bacteria and archaea, have the cell type or superkingdom prokaryotes, and the eukarya have the cell type or superkingdom eukaryotes. Some characteristics of the domain bacteria: all the cell types are prokaryotes. If they have a cell wall, they all have peptidoglycan in their cell wall. Most of the domain bacteria multiply by binary fission. That's where the cell literally splits into two and becomes two cells. One cell splits into two and becomes two cells. For energy use, the cells can obtain their energy needs from organic chemicals, such as glucose, and we can do that, from inorganic chemicals, such as iron, hydrogen sulfide, sulfate, nitrate, nitrite, etc. Uh, the chemosynthetic bacteria get their chemical needs from an inorganic chemical. We'll talk a little bit about them. Or they can get their needs from photosynthesis. And the bacteria can get their energy need from all the available energy sources. Most of the domain are free-living decomposers or primary producers, but some of the domain bacteria are parasitic and some are pathogenic. Remember, a pathogen is a microbe or a worm which can cause disease. Any question about the domain bacteria? If not, let's move on to the domain archaea. They are cell type prokaryote. Does anyone know, do the domain archaea, do any of the species have peptidoglycan in them? Anyone know? We'll come back to that so you can think about it. It's either yes or no if anyone wants to take a guess. But some of the archaea are found in the extreme environments, and that was actually the first place where we found archaea. However, when we look further, we find that the archaea are found in all of Earth's ecosystems. They actually make up a significant portion of the microbes in the soil and the microbes in the plankton in the ocean. They include the methanogens, the extreme halophiles, the extreme thermophiles, and these are the extreme environments we find them in. But like I said, we find them in all of the Earth's biomes. Here's another question for you, and that is, are any of the members of archaea pathogenic, meaning causing disease in humans? Anyone know? Anyone want to guess?
No.
You're correct. None of the archaea are pathogenic. So for this reason, we're not going to be focusing on the archaea, and I hardly will discuss them in this class, other than in this lesson here. And then we do not find peptidoglycan in members of archaea. Peptidoglycan is only found in members of bacteria if the bacteria has a cell wall. Talking about the eukarya, and I've already mentioned, all of the eukarya lack peptidoglycan. We can split the eukarya into 4 kingdoms: kingdom plantae, the plants; kingdom animalia, the animals; kingdom fungi, the fungus; and kingdom protista, the protists. Remember, the protista are a non-monophyletic group, meaning they are a collection of eukaryotes that are not plants, not animals, not fungi. And we're just lumping them all together in the kingdom protista. For the kingdom fungi, you don't really need to know they have chitin in their cell walls, but they use organic chemicals for their energy needs. That's the same as us. We use organic chemicals for our energy needs, like glucose. The fungi have multicellular molds and multicellular mushrooms, as well as the unicellular yeast. The mushrooms are composed of mycelia, which are composed of filaments called hyphae. And here we're seeing some hyphae here. I think that's a mold, not a mushroom, but. And there's some unicellular yeast. I think you're all familiar with yeast. We put it in our bread to make bread, and at least leaven bread. And it can cause disease like I'm drawing a blank there on what the heck that name is. Well, it can cause yeast infections, usually from Candida albicans, that yeast. So the fungi, most of them are decomposers or pathogens, and in fact, all of them are decomposers or pathogens. The kingdom protista include plant-like protists, and that's the algae, the protozoa, or the animal-like protista, and then the fungus-like protista. The algae can engage in photosynthesis. They have cellulose in their cell walls. You don't really need to know that, but they do produce molecular oxygen and organic compounds like glucose. The protozoa include amoeba, paramecium, and euglena. They absorb and ingest organic chemicals. They may be motile, but more than just flagella, bacteria can only move, as well as archaea, can only move if they have flagella, or at least one flagellum. But the protozoa can move if they have flagella, or they have cilia, or they have pseudopods. Some of the protozoa are parasites, like Entamoeba histolytica and amoeba, malaria, which is a protozoa. Any questions about the algae or the protozoa? We also have some protists which are fungus-like. I'm not really going to talk much about them. The only one I'm going to mention is the potato blight is actually a fungus-like protist. What caused the Irish famine, I don't remember when it was, like the late 1800s, I think, in Ireland, which went through the potato crop, ruining the potatoes of Ireland, which made the Irish starve, and so a lot of Irish came to America at that point, was caused by a fungus-like protist. All right, let's talk briefly about the kingdom Plantae. We can divide the plants into mosses, ferns, conifers, and flowering plants. All the plants are multicellular. There's no unicellular plant. I should say with the protists that some of them can be single-celled, like all the protozoa are single-celled, but some of the algae can be multicellular, like the algae that washes up on the Oregon or Washington beaches. That's multicellular, but you can have single-cell algae, as well as the Volvox, which is a multicell, but it's small enough that you need a microscope to see it. All the plants are multicellular, and there are no plants that cause human disease. So we're not really going to study plants in this class. Plants, most of them are green and can engage in photosynthesis, but not all plants are green, and not all plants, the ones that are not green, do not engage in photosynthesis. For your information, you don't need to know this for the class, but the non-green plants are actually parasites, parasitic plants that parasitize photosynthetic plants. All right, let's move on to the kingdom animals. Animals include the sponges, the worms, scientific name helminths, the insects, and the vertebrates. All of the animals are multicellular. Any question about any of that? We will talk a little bit about the animals in this class because the worms, helminths, can cause human disease. Parasitic worms. All right, we've talked a little bit about microorganisms, and we mentioned that a microbe or a microorganism includes bacteria, archaea, which have cells, and viruses and prions, which are not organisms because they do not have any cells. They do include certain eukarya, all the protozoa, molds, and yeast are microorganisms. Some of the algae are microbes, and then it also includes the helminths, or the worms. But most worms are not microscopic, correct? Like the beef tapeworm, I don't remember if it's 30 feet or 30 meters, it's something like that. It's huge, at least the fully adult, fully grown adult. But the helminths are considered microbes because even though the adults are multicellular animals, many of the helminths are parasitic, and they're either in the flatworm or the roundworm category. All of the earthworms are not parasitic, so we will be studying some flatworms and roundworms because they cause human disease. The adults' worms are not strictly microbes, at least most of them, but some of the life stages of the worms are microscopic, including the stage of the worm which is infective to humans. The infective stage is always microscopic. Is that true? There might be a worm which can actually burrow through your skin. Of course, that's a microscopic burrower. So, as far as I know, all of the helminths, um, the infective stage is microscopic, even if the adult is a huge worm. There are a few worms which are, even the adults are microscopic, but most are huge. All right, let's talk just briefly about viruses. They are cellular. They do consist of DNA as their core molecule, meaning their genome is either DNA or RNA. It is never both. The core is surrounded by a protein coat. The coat may be enclosed in a lipid envelope, but not all viruses have this lipid envelope. Viruses only replicate when they've infected their host cells. Outside of their host cell, a virus has no metabolic activity and they do not replicate. Viruses are not part of the three-domain system, mainly because they don't have cells. Any question about viruses? We'll talk more about viruses when we talk about chapter 13 at the end of this class. So looking at the three domains of cellular organisms, we have domain bacteria. Let me blow this slide up. We have domain archaea and domain eukaryotes, cell type for bacteria and archaea, prokaryotes, cell type for eukaryotes, eukaryote. When we look at the chromosome of bacteria, there's usually one, and it's usually circular. No histones on that chromosome. For archaea, there's usually one chromosome. It's usually circular, but it does have histones on it. A histone is just a protein that you usually find in eukaryote DNA, but you also find it in archaea DNA. Eukaryotes have a different chromosome structure, and that is there's many of them, not one. The chromosome is not linear, meaning it does not end where the chromosome begins. It's usually a linear chromosome, like, oops, trying to draw the chromosome X like this. The X has a beginning point here and an ending point here. It's not a linear chromosome. All of the DNA in eukaryotes is wrapped around histone proteins. When we look at the ribosomes of bacteria and archaea, they are the same, 70S in size, but the eukaryotes have a larger ribosome, 80S in size. The S just stands for the sedimentation coefficient, where the ribosome sediments on an ultracentrifuge gradient. And then the domain bacteria contains peptidoglycan. If the bacteria has a cell wall, it will have peptidoglycan as its main molecule. The archaea has no peptidoglycan, and the eukaryotes have no peptidoglycan. Any question about this comparison of the three domains? If not, let's talk about a brief history of microbiology. When we're talking about the history of microbiology, you should come to understand that it largely concerned a debate over whether microbes could spontaneously generate or whether they could only come from a pre-existing life form called the theory of biogenesis. The history of microbiology also encompasses the golden age of microbiology. You don't need to know the timeline in the mid to late 1800s to the early 1900s, where when microbiologists looked, everything they discovered was something new that somebody hadn't known before. This is the golden age of microbiology. And this is showing you the history of microbiology, the findings before microbiology was started, the golden age of microbiology, and then the history of microbiology after the golden age. We'll discuss some of these. When I'm going through the history of microbiology, you don't need to know any of the dates. I've just got the dates here to anchor it, except for the first date. You should know that 3.5 billion years ago or earlier, we can find ancestors in the fossil record of the first bacteria. And this is the first life we found on the planet Earth. There's an actual fossil, and there's the artist's rendition of it right there. So 3.5 billion years ago or older, we have the first ancestors of bacteria in the rock of the fossil record. The next event is the Babylonians wrote down how to make beer recipes, and they didn't know it, but they're actually using microbes to make the beer. And then the next record is the Mayans made a fermented beverage from cacao that we would call chocolate. They didn't actually have milk in it, but it was close to hot chocolate. It's just that it wasn't, I don't think it was hot, and it really wasn't chocolate because it was in water, and they tended to put like peppers in to season it as well. But it would be close to chocolate. The only thing you need to know is that these three events happened, and in this order, you first had to have the fossil record having the first living organisms, and then the first written record of a recipe for using microbes to make beer, and then the written records of the Mayans for making chocolate, a fermented beverage from cacao. Any question about any of that? So you do not need to know the time for anything except for this first one. Our next event was the invention of the microscope, which you don't need to know that it was invented in 1590 by Janssen, but we had to have the microscope invented before we could actually see microorganisms. Robert Hooke was actually the first person who used the microscope to report on something alive that he looked at. And what he looked at was, let me end the show here. What he looked at was shavings of cork, which is actually what we put in a bottle of wine to cork it, and it's actually using, we call it a cork plug, and it plugs the bottle of wine, but it's actually from the cork, which is sort of the bark of the cork tree. So cork is actually used three ways here. And what he did was he took thin shavings of the cork tree, or the bark of the cork tree, and looked at it under the microscope, and what he saw, let's see if I can get it here. There we go. A little slow. What he saw was actually things that he called cells. Oops, that's not very straight. Try and make that straight. Imagine it straight. Boy, that didn't even write there. I'm not doing a very good job of making this straight. And he called it cells because in a monastery, the monks would have what do you call that, hallway, and off the hallway there would be a door here going into the monk's private room, which they did whatever monks did in their private room, and they called this a cell. And so the cell of a living organism is named after the private room in the monastery of the monks. Any question about any of that? You don't need to know the year, but after Robert Hooke reported this, finding little boxes or cells in plants that he called cells, this marked the beginning of the cell theory, the theory that all living things are composed of cells. This theory was further developed and actually wrote up by Virchow, that all living things come from cells. The above here are not specifically microbiology, the invention of the microscope, and even Robert Hooke's looking at cells of plants was not strictly microbiology. Oh, let me state that Robert Hooke, when he looked at tissue of animals, he did not see any cells. And the reason being was the early microscopes were blurry, and when you looked at the tissue of animals, you could not see cells. The reason being is that animals lack a cell wall, and plants have a cell wall, and the cell wall is very thick and rigid. And that's why Robert Hooke, when he looked at the tissue of plants, he could see cells. But when he looked at the tissue of animals, he could not see cells because cells of animals only are separated by the cell membrane, which is much thinner than the cell wall of plants. Any question about any of that? After the microscopes improved, which probably was 100 or 200 years later, people then started looking at animal tissue, and they could focus in on the cells good enough that they could start to see cells in animal tissue. But they did not see it initially, and I'm guessing it was at least 100 years, maybe even 200 years. All right. At about this time, there were different hypotheses about how microbes could originate. And actually, initially, uh, people suggested, or scientists suggested, that life could originate from one of two competing hypotheses. And that is, one, spontaneous generation of life could arise, where when a vital force comes into inorganic matter, it can change the inorganic matter to give rise to life spontaneously, naming spontaneous generation. And the other theory is that living organisms could only come from other life forms. And that's the theory of biogenesis, that all life arises from life. Now, even in the earliest days, everyone would agree that a baby, a complex life form, had to come from its parents So babies came from living organisms. However, simpler life forms, people argue, could spontaneously generate. So over a century ago, there were beliefs that a newt could arise out of a log when you put a log in the fire. In reality, the newt was probably in the bark of the log, and you put the log in the fire, the newt felt the heat, and then crawled out of the fire to escape the heat. But back over 100 years ago, people would see the newt coming out of the fire and they'd say, spontaneous generation of the newt. And then people would see flies emerging from manure and they would say, spontaneous generation of flies. People would also see maggots arise from a decaying corpse and they'd say, spontaneous generation of the maggots. Most were logical, none were scientific, and some were even amusing. Somebody wrote up that if you put sweaty underwear in a jar of wheat and you put wheat kernels in this jar, that would spontaneously give rise to the mice. In the reality, the mice climbed into the jar to get at the wheat and then eat the wheat. And then they couldn't get out of the jar because they couldn't climb up the glass of the jar. And the sweaty underwear actually had nothing to do with the mice. Any question about any of that? A brief sidetrack. When we were talking about microbes, in the early days, we'd have a beaker of media like this. Let me blow that up so you can see it a little better. And it would be clear. And then over time, even if you capped it, it would become cloudy like that, swarming with microbes. And many people believe that microbes spontaneously generated because we always saw this happening, even if you cover the dish, even if you poured sterile media into the beaker and then covered it, you would get this happening. And that's because the container had microbes in it to begin with. The first evidence looking at whether life could evolve spontaneously or whether it was generated was done by Francesco Redi. And you should know Francesco Redi because not only did he provide the first evidence against spontaneous generation, he also wrote up the first documented controlled scientific experiment where he wrote up an experiment where he actually controlled the variables of the experiment. And that is, he had 3 jars completely sealed and 3 open jars, and he found maggots only in the open jars, not in the sealed jars. Well, the people who believed in spontaneous generation said, no, Reddy, you didn't allow the vital force to get in the jar. You completely sealed the jar. So the vital force couldn't get in. So that's why there are no maggots. And Francesco Redi made an ingenious experiment where he had an open jar, covered it with a fine wire net, and noticed that the jars of meat had no maggots in it, even though it was open for the vital force to come in. This did not support spontaneous generation, and after Francesco Redi's experiment, everyone agreed that complex life forms could only arise from pre-existing life forms. The complex life forms, like a maggot, could not arise spontaneously. However, people still argued that microbes could spontaneously generate. And part of the reason is that when you covered the meat with the wire mesh, you could see microbes growing in the meat. I'll skip that. And about this time, we have the first birth of microbiology, where the first microbes were actually observed by Antony van Leeuwenhoek. I'm not Dutch, so I could be mispronouncing his name. and here he is here. He described live microorganisms essentially any place he looked, like on scrapings from his teeth, in rainwater, in peppercorn infusions, as well as he sampled the River Thames and found it loaded with microbes. And the British weren't too happy about that, especially the Londoners, because they were drinking the water from the River Thames as their drinking water. This is actually a microscope that he built, and he was an amateur microbiologist and an amateur microscope builder as well as a lens grinder. He ground his own lenses. And he was such a good grinder that he was one of the best lens grinders of his day. His microscope is simpler than, um, um, what was it, Jenner's microscope, who had a compound microscope. And Tony van Leeuwenhoek only had one lens, a simple microscope, which is essentially a super duper magnifying glass. But because he was only using one lens and did the lens grinding himself, he was able to finely focus with his microscope. And he was able to see things, cells, so small that they were not seen for another 100 years until the microscope got better, and then they could actually finely focus and see the microbes that Tony van Leeuwenhoek saw a century earlier. And here he has a drawing here, which are actually bacteria. There's a rod, there's the movement of the rod, there's a sporillum, there in figure E are two cocci, and then here are rods, that's the bent rod. So he's actually drawing bacteria, cells that were so small they were not seen by other microbiologists for another 100 years. Any question about any of that? All right. And then Rudolf Virchow proposed that all cells come from pre-existing cells. Many actually wrote it up in 1858. But arguments about spontaneous generation continued in 1861, meaning people still argued up until 1861 that microbes could spontaneously generate. What happened in 1861 was this guy, Louis Pasteur, finally made an experiment disproving the theory of spontaneous generation and therefore confirming the theory of biogenesis. How he did this was he demonstrated that microorganisms are present in the air and also in the tube, and you have to destroy those microbes before you can put the media in, sterilize the media, and put the media in, and then you will not get microbes growing in the tube, assuming you seal the tube. You have to protect it from the air, but you can use heat to sterilize the microbes, both in the air and in the tube. And then he made his ingenious, famous swan neck flax experiment to disprove the theory of spontaneous generation. What he did was he had a beaker with a long neck like that, poured the media in the beaker, and then he melted the flask here, bending it into a swan neck, sort of an S-type shape there. And then he heated the media to kill the microbes in the media and in the glass tube beaker. And then he let it sit, and there were no microbes in the media. He then showed that if you take the media and bring it down to here, don't put it up to there, but actually bring it down to here, and then put the media back, you can get microbes growing in the media. What he was showing was that microbes were falling out of the air, but in the swan neck, they got here because they don't come up against gravity. and that would keep the flask free of microbes, showing that spontaneous generation of microbes does not occur, and this flask is actually open to the outside so the vital force can get in. Any questions about Louis Pasteur's experiment disproving the theory of spontaneous generation? All right. Beginning with Pasteur's work, people began wondering about the relationship between microbes and disease. This later became known as the germ theory of disease. We later got out of that immunity and the development of antimicrobial drugs, drugs that we could use against germs that cause disease. There was no one author of the germ theory of disease, meaning many people proposed it over time, and they developed it. One person said maybe it causes this one disease, and then other people said, well, it can cause these other diseases. Around this time, you don't need to know the dates here, but Pasteur developed the basic idea of aseptic technique. That's important with working with microbes so that you could work with a pure culture and then keep it pure. About this time, also Koch developed the technique of streaking for isolation of colonies on a solid media, which allowed us to develop a simple way of getting a pure culture. Louis Pasteur came up with a way of getting a pure culture, but it was a very difficult method. And Edward Koch, or Dr. Koch, came up with a simple method of getting a pure culture. And that is you simply streak out the cells 2 times through a colony forming event. And if you do it 2 times, it's a high probability that the resulting cells are from one species. So you get a pure culture. About this time, Hess, somebody working in Dr. Koch's lab, developed a nutrient auger. It was a new medium, a general purpose media for growing many microbes. Before that, people were growing it on gelatin or something else. And the trouble with that was that sometimes the microbes could digest the gelatin and make a mess. People were growing it in liquid culture like Louis Pasteur before that, but it was difficult to obtain a pure culture. Not impossible, but difficult. And about this time, Winogradsky developed the idea that microorganisms were involved in the biogeochemical cycle, such as the nitrogen cycle and the sulfur cycle. Well, with Pasteur's work, and then the discoveries between the relationship of microbes and disease, the germ theory of disease developed, and that is that infectious diseases are caused by germs, microorganisms, which cause a disease. Out of that work came immunity and the development of antimicrobial drugs. Pasteur showed that microbes are responsible for fermentation, that the microbes were fermenting the sugar in wine, for example, converting grape juice into alcohol or grape juice into wine. And that happens in alcoholic fermentation, conversion of sugar to alcohol, and it's used to make beer and wine. Microbial growth is also responsible for the spoilage of food. You should realize that bacteria could spoil alcohol by digesting it and then converting the alcohol into acetic acid, spoiling the wine and converting wine into vinegar, acetic acid, by fermentation. Any question about any of this? All right. Pasteur showed that the spoilage bacteria could be killed by heat, and you could do this by even using heat not hot enough to evaporate all of the alcohol in wine. And so Pasteur developed pasteurization, an application of a high heat for a short period of time. It's used in a variety of foods. Pasteur initially developed it for beer. And what you do is you heat the beer for a short period of time, killing the spoilage organisms as well as all human pathogens, but not high enough heat that it totally evaporates the alcohol in the beer. Any questions about any of that? Well, from fermentation, the realization that microbes can affect organic matter, changing sugar into alcohol, ethanol, people began asking, could microbes have a similar relationship with plants and animals? Microorganisms cause disease. This concept became known as the germ theory of disease. This is important to understand that microbes cause disease because before this came out, the general belief was that poisonous vapors were breathed in, and then the poisonous vapors cause disease. Like when somebody got cholera from walking through the swamp, it would assume that the poisonous vapors in the swamp were breathed in, and then the person developed cholera. In reality, people walking through the swamp can be exposed to the organism which causes cholera. It can breathe it in, and then it can cause cholera in the patient once it gets established. Any question about any of that? All right. From the germ theory of disease, we had the concept that organisms could cause disease. The first person to actually show that was Ignaz Semmelweis, and I'm not German or Austrian, so I could be butchering his name. But Semmelweis advocated hand washing to prevent the transmission of postpartum and puerperal fever between OB-GYN patients. Before that time, doctors would perhaps go through surgery on a body and then go and visit pregnant females, and they could cause postpartum fever. Any question about Semmelweis? Sad news in Semmelweis was he was requiring the doctors in his department, which was the OB-GYN department, to wash their hands before they went to a patient. And it actually, he showed significantly that it did reduce the infective rate of the patients. He proposed that all surgeons do it as well as other people in the hospital, but the president of the hospital, who was a doctor, was so insulted that he actually fired Semmelweis for proposing that doctors wash their hands and then requiring it in his own department. Any question about that? All right. The next person who showed the germ theory of disease and that how to prevent getting disease was Joseph Lister, who used a chemical disinfectant to prevent surgical wounds. What he did was he used carbolic acid, which we call phenol, to treat a patient's skin before engaging in surgery. And then he also sprayed it on the surgical instruments. So he reduced infection from surgery. In the old days, when they did surgery, at least 50% of the patients usually died of infection. And after Joseph Lister, he dramatically reduced the surgical infections and greatly improving the survivability of the patient undergoing surgery. One problem with Joseph Lister is he was using phenol, which is corrosive to metal and to human skin. So, yes, he reduced the surgical infections causing death, but he was treating the patients with something which would be unpleasant, and he didn't know it at the time, but phenol also increases the risk of cancer. So, as you can imagine, we no longer use phenol, but we've come up with better agents to use than phenol. In Joseph Lister's day, when he tested various chemicals, the one that had the most antimicrobial property was phenol. All right, any question about Joseph Lister? Come on. All right, about this time, Koch actually proved that a bacterium could cause a given bacterial disease. And what he did was he showed that the Bacillus anthracis bacteria could cause anthrax, the disease anthrax. And then Koch also provided the experimental steps required to prove that a specific microbe causes a specific disease. This became known as Koch's postulates, the best way to show that a given microbe causes a given disease. So actually, Dr. Koch finally proved the germ theory of disease. And that's because he showed that Bacillus anthracis can cause the disease anthrax. All right, any question about any of that? Near this time, we actually had the development of our first vaccine. Edward Jenner, in 1796, you don't need to know the year, developed the first vaccine against smallpox. How we made his vaccine was he simply used someone who had cowpox, scraped the infection of cowpox from the patient, and then used it against patients to prevent their getting smallpox. The person was protected against getting smallpox, but the means for this vaccination were not understood in Edward Jenner's day. And it was not until Louis Pasteur came around and experimented with making vaccines that he finally understood what was going on. Cowpox is genetically related to smallpox. When somebody makes an immune response to cowpox, some of that immunity can cross react and attack smallpox, meaning an antibody. Let's say there were 10 antibodies made against cowpox, and at least one of those antibodies, besides binding to cowpox, also bound to smallpox. So that cowpox was working to prevent getting smallpox simply because cowpox had a genome similar to smallpox. And then when we put that genome into the patient can then mount an immune response against it. And then the patient's immune system is now primed and ready to go. So the next time the person gets exposed to smallpox, they will be immune to it. Now, let me state that Edward Jenner really only had his own intuition to go by. when he first injected two boys, one being his son. The first boy injected was James Phipps. And he didn't know what was going to happen when he injected cowpox into the patient, but he expected they would just come down with cowpox, a very mild disease containing maybe one pustule on the skin, meaning not many like smallpox, and that the patient might have an immune system response and go into like a headache. And actually, James Phipps did have, I think, most of those side effects. But then, after everything looked good and the smallpox was coming through the villages in England, Dr. Jenner actually injected the smallpox organism or viruses into Phipps, and then later saw that Phipps did not come down with smallpox, proving that he was protected by the vaccine against which was actually cowpox. Any question about any of that? All right. Like I said, what was happening when this was first discovered by Pasteur was that the genome of cowpox was similar enough that in an immune response, like against one antibody, cross-reacted to an antibody on the smallpox bacteria, making the patient immune. The term vaccination is actually derived from the word vacca, and that's because the first vaccine was cowpox, a disease coming from vacca. Vacca means cow, by the way, if you don't know. The protection you get from a vaccine is called immunity. Any question about any of this? All right. Chemotherapy is the treatment of a disease with a chemical. Chemotherapeutic agents for an infectious disease are sometimes called synthetic drugs or antibiotics. But in the old term, we did call it a chemotherapeutic agent. Today, chemotherapy is referred to almost exclusively for a drug to treat cancer. But textbook definition means it's treat any disease. And in older times, it was used to treat a bacterial caused infection. Antibiotics are a chemical produced by a living organism that inhibit or kill other microbes. This is the textbook definition for an antibiotic. That's not the way the term antibiotic is used in the general public, or the way it's used, at least in most clinics, meaning doctors themselves tend to use the word antibiotic on any drug that can be used against a bacteria causing an infection. And this is somewhat simpler if you use the term antibiotic that way, because if you use antibiotic with the textbook definition, penicillin G is an antibiotic because it was made by a microorganism, a living species. But carboxylin is not an antibiotic because it is a man-made antibiotic. So it's antimicrobial, but it's not a, strictly speaking, antibiotic. Any question about any of that? I will use the term antibiotic as is used in the general population, meaning antibiotic is any drug capable of being used on a bacteria to prevent or to cure an infection. Paul Ehrlich found the first drug to treat syphilis in 1910. He was looking for the magic bullet, a drug which could prevent well, which could cure a patient from a bacterial infection. Curiously, the first chemotherapeutic agent was not one that Paul Ehrlich found. It was actually quinine from the bark of the cinchona or the cinchona tree, and it had quinine in the bark. And if you ate this bark or consumed it, you would be able to throw off malaria because quinine is an anti-malaria drug. Anyone want to guess when quinine first became available in Europe? Just
Was it
guess.
recent?
Say again?
Was it recent?
No, because we treat malaria nowadays with quinine and what is it, chloroquine and other anti-malaria drugs, but we make them
Is
today,
it
so it's not recent. Well, was it before or after 1910?
before?
Before. How long before?
I don't know. I'd say maybe like 100 years.
That's a good guess, but no, it's not correct. I'm just pointing out to you the first chemotherapeutic agent which was used in Europe against malaria was actually available in the mid 1600s. What happened was Jesuit missionaries went to Peru and they saw that natives of Peru were eating the bark of this tree to cure their malaria. And they saw it was effective, meaning the patient no longer had malaria. And then they took the bark of this tree and made it available to Europeans. And that became the first known treatment of any disease being cured by the bark of the cinchona tree. Who knows when the Indians, the natives of Peru, discovered that the bark of this tree could be used to prevent malaria. But like I said, in the mid 1600s, we had an anti-malarial drug, which was way before Paul Ehrlich came up with the magic bullet. Let me briefly talk about, um, well, chemotherapeutic agents, and that is in 1910, Paul Ehrlich came up with a man-made arsenic drug which could cure syphilis. And then Alexander Fleming in 1928, you don't need to know the name, but you should know Alexander Fleming, discovered the first antibiotic. What he did was he streaked out bacteria on a plate, and Fleming was a sloppy microbiologist, and a mold spore landed on the plate. And then Alexander Fleming took off for the weekend, leaving the plate on the counter. If he had put the plate in someplace colder or someplace warmer, like his incubator, he would not have gotten two things growing on the plate. As it was, he got two things growing on the plate, and the plate looked something like this, where he had the streak of bacteria, and then he had a colony of mold, and he noticed around the colony there was a region of inhibition where the bacteria was not growing. Alexander Fleming correctly concluded that something was coming out of the mold and preventing the growth of the bacteria. And he called this penicillin, specifically penicillin G, which is an antibiotic made by the mold Penicillium. So it's a penicillium mold, making the antibiotic penicillin as the first antibiotic discovered. Unfortunately, Alexander Fleming could not purify his penicillin, so you could not use it on a patient. The only thing he could use would be to grow it in media, remove the cells from the media, and then use the pure media, which obviously you couldn't give that to a patient. Certainly couldn't inject it into the patient. Maybe they could drink it, but it wouldn't taste very good. The first clinically available antibiotic was sulfonamide, a sulfa drug, which was synthesized, you don't need to know this, 1935, but it became the first clinically available antibiotic. The reason being is that humans were able to make this drug, and so it was fairly simple to purify, and then it became clinically available. Penicillin did not become clinically available in the 1940s, so not until World War II, where it was tested clinically on 2 patients, mass produced, and then they found penicillin G actually works. If there's ever time, you can ask me about the story for the scientists who studied the first 2 patients that they were treating with penicillin G. But there isn't time today. All right, any question about any of that? If not, I'm going to end the class here, and we will continue with chapter 1 next time. I'll see you at 6:30 in the lab unless there's a question. All right, I'll say goodbye.